In this lecture, we'll look at the earliest stages of visual processing, beginning with a brief examination of some of the physical properties of light. In the second part, we'll begin our investigation into the eye. In the third part, we'll look at photoreception, the rods and the cones. These are the cells responsible for the transduction of light energy into neural energy. In the fourth part, we'll have a look at post-receptoral mechanisms, before finally turning our attention to receptive fields. Like all sensory systems, vision relies on environmental input. In the case of vision, this environmental input is light. When we perceive a visual object, we refer to the light being transmitted by this object as the distal stimulus. This light, which has entered the eye and is projected onto the retina at the back of the eyeball, forms an image. This projected retinal image is known as the proximal stimulus. This light, which constitutes the proximal stimulus, is first converted into neural energy by photoreceptors, known as rods and cones. This neural information is then transmitted to a dense network of neurons within the retina, known collectively as post-receptoral mechanisms. This information is then transmitted out of the eye and into the brain, where it's subjected to further neural processing. Some of this visual processing that occurs within the brain manifests as visual perception. Light is electromagnetic energy which is being transmitted throughout the universe and whose primary source is stars. On Earth, the light that we see represents but a mere fraction of the solar radiation which manages to make it through our atmosphere. Light is massless energy. This energy travels in discrete packets called photons. Photons can be thought of as either particles or waves, depending on how they're measured. The amount of energy within a given photon is reflected in the wavelength or frequency structure of its wave. Higher energy states have shorter wavelengths than low energy states, which have longer wavelengths. Most wavelengths are invisible to the human eye. Humans are sensitive to a narrow band of light, with wavelengths between about 400 and 700 nanometers. 1 nanometer corresponds to a billionth of a meter. That white light is composed of multiple wavelengths, which we experience as colours, was first demonstrated by Sir Isaac Newton, who showed that when white light is passed through a prism, it fans out into a rainbow, showing its constituent wavelengths or colours. Most of the light that we see when we interact with the world does not come directly from light sources, but instead is reflected from surfaces in the environment. Exceptions to this occur when we see light sources directly. In the natural world, this is usually the sun or more distant stars. It can also include artificial light, such as illumination from light bulbs, TVs, or smartphones. One of the more remarkable aspects of vision is that we have a perception of objects in three-dimensional space, although the distal stimulus may be a three-dimensional object. The proximal stimulus, that is, the stimulus or the image that's projected onto the retina, is actually a 2D surface. Somehow we're able to extract or infer 3 dimensions from just 2-dimensional proximal input. Part 2. The eye. When light enters the eye, it first passes through a transparent structure called the cornea. This causes the light to bend or to refract by a fixed amount. Most of the focusing that occurs within the eye occurs in the cornea. Light then passes through a small aperture called the pupil. The pupil size is under the control of the iris, the muscular structure which gives our eyes their colour. The pupil size varies in response to numerous factors, such as the total amount of light entering the eye, but also higher level factors involving cognition and arousal. After passing through the pupil, the light is further refracted by the lens. The lens's curvature and hence its refractive index can change. Changing the lens's curvature has the effect of modifying how focused the image will appear at the back of the eye. Changing the shape of the lens is known as accommodation. Accommodation has the effect of bringing objects at different distances into focus. The retina refers to the dense network of neurons that cover the back of each eye, and upon which environmental light is projected. The retina is composed of photoreceptors, which are the cells responsible for the transduction of environmental light energy into neural energy, as well as post-receptoral mechanisms, including horizontal cells, bipolar cells, amacrine cells, and ganglion cells, the gatekeepers of the retina. Each eye's retina contains a small region known as the fovea. When we look at an object, that is to say, when we fixate on it, we invariably place that image onto our fovea. This is because the fovea is the region of retina which affords us the highest spatial resolution. The fovea represents a relatively tiny region of the total visual field, less than about 5%. The area outside the fovea is called peripheral vision. The further away a proximal stimulus image is from fixation, the larger it will need to be to appear equally focused as the same object presented at fixation. This demonstrates peripheral vision's relatively poor spatial resolution. Interestingly, even though visual resolution varies quite dramatically across the visual field, one doesn't generally feel as though peripheral vision is out of focus, but it is. You can easily demonstrate this for yourself. Try looking at a specific object, such as one of the letters on this slide in front of you. While you're doing that, try to identify an object or a word in your peripheral vision. I'm sure you'll struggle. This begs the question then of why does peripheral vision afford us poorer resolution than our central vision? To answer this question, we must first look at the photoreceptors. There are two primary classes of photoreceptors, rods and cones, which describe differences in their morphology. But there are anatomical and functional differences as well. Whereas rods cover the entire visual field, except the fovea, cones are more densely packed in the fovea. Functionally, rods are highly sensitive to low illumination and are consequently most useful during night vision. Cones, by contrast, are sensitive to high illumination and are therefore used for day vision. Another functional distinction is their role in colour vision, whereas cones, which come in three types, are sensitive to colour variation. Rods, by contrast, are insensitive to colour variation. There are three classes of cone: short wavelength sensitive cones, middle wavelength sensitive cones, and long wavelength sensitive cones. Shortwave light approximates our experience of the colour blue or violet. Middle wavelengths approximate our experience of the colour green, and long wavelengths approximate our experience of the colour red. There are approximately 6 million cones in each eye. In the case of rods, these are far more numerous, and as I said, they're not located in the fovea. This variation in the relative density of rods and cones can be seen graphically in the figure on the left. The x-axis, labelled eccentricity, represents a single lateral cross-section through the left eyeball. Eccentricity refers to the distance from the point of fixation, that is, the eye's fovea. Consequently, zero eccentricity represents the fovea. Increasing eccentricity implies an image being projected further into the periphery. The numbers to the left of zero eccentricity, labelled temporal, represent the half of the retina closest to the left ear. The numbers to the right of zero, labelled nasal, represent the half of the retina closest to the nose. The graph's y-axis describes photoreceptor density. There are a few features of this graph I'd like you to notice. The first is that density of cones, indicated by the red curve, is sharply peaked in the fovea and falls off dramatically as one moves into the periphery. By contrast, the rods, represented by the blue curve, are absent in the fovea, but far more numerous than cones in the periphery. This has several functional implications for how we see. The first of these is colour vision. Because the cones, which, as we've mentioned, are responsible for our colour vision, are relatively underrepresented in the periphery, our perception of colour is relatively poor in our peripheral vision. Another implication involves night vision. Have you ever noticed when looking at the night sky that it's very difficult to see dim stars when you look directly at them? It's in fact easier to spot them if one looks just to the side of the star. This is because the receptors that are most sensitive to dim light are the rods, and these are absent in the fovea. Another functional implication of these differences in cone and rod density is visual acuity. The cones, which are functionally useful in daylight conditions, are underrepresented in the periphery, meaning we're simply less efficient at sampling peripheral vision than we are images on the fovea. The final component of this figure that I'd like you to notice is the vertical white stripe, which is labelled here as the optic disc. The optic disc is the region of the retina where there are no photoreceptors. The reason for this is that this is where the retina's neural fibres are bundled together and leave the eye to connect with the rest of the brain. A curious consequence of this is that we actually have a blind spot in each eyeball. You can experience this blind spot for yourself using the following demonstration. Cover your left eye and fixate on the white cross in the centre of the screen. Now, stretch your arm out towards the screen, careful not to cause damage, and raise your thumb so that it's just to the right of the cross. Keep fixating on the cross. While you're doing this, slowly move your thumb to the right. What you'll notice eventually is that there comes a point where the thumb will simply disappear from view, probably when it's about 15 centimeters to the right of the cross. Keep moving your thumb to the right and it will reappear. You've just experienced your right eye's blind spot. What's perhaps most remarkable about this is that although you possess two blind spots, one in each eye, one rarely, if ever, notices them. The visual information around each blind spot seems to perceptually fill in. The back of the eye is where the photoreceptors, that is, the rods and the cones, are located. Once a photoreceptor absorbs a sufficient number of photons, its neural signals then propagate to the post-receptoral neurons, that is, the horizontal, the bipolar, the amacrine and the ganglion cells. The ganglion cells represent the final stage of retinal processing. If ganglion cells don't respond, the response to a given image will not make it out of the eye. In this sense, we can think of the ganglion cells as retinal gatekeepers. Not all ganglion cells are wired up to their photoreceptor inputs in the same way. Some ganglion cells receive inputs from a large number of photoreceptors. This is common in the visual periphery. Other ganglion cells receive inputs from a single or just a few photoreceptors. This is more common in the fovea. This difference in the amount of neural convergence has functional implications. When many photoreceptors converge onto a single ganglion cell, this ganglion cell may be more likely to respond than a ganglion cell which receives input from just one photoreceptor. This increase in sensitivity comes at a cost, however. When the amount of convergence is high, the ganglion cell cannot distinguish which of its many photoreceptor inputs was stimulated. Consequently, information and resolution is lost. Conversely, when the amount of convergence is lower, less information is lost and resolution is greater. Let's imagine that we're presenting two short yellow lines to the fovea and two short yellow lines to the periphery. Because there's less convergence in the fovea, each line will stimulate a unique ganglion cell. If only one line was presented, only one ganglion cell would be stimulated. In other words, the brain would be able to tell which line or whether both lines were presented, and no information would be lost. Now let us turn our attention to the periphery. Because both lines are capable of stimulating a single ganglion cell, that ganglion cell would not be able to tell whether one line was presented or both. Information would be lost. Ganglion cells don't just receive excitatory photoreceptor inputs. In fact, they respond to specific patterns of light. On the right, we have a wiring diagram. The circles at the top, labelled R, represent photoreceptors. The receptors located in the central disc above, labelled the ON area, feed into a bipolar cell. The receptors located on the outer ring, labelled the OFF area, feed into horizontal cells. Now, if this bipolar cell is stimulated, it will drive the ganglion cell. Importantly, however, the horizontal cells send inhibitory input into the bipolar cell. If inhibited, the bipolar cell will not fire, and nor will the ganglion cell. To make this bipolar cell fire, it must receive greater excitatory input from receptors in the ON area than in the OFF area. If the receptors in the OFF area are stimulated, this will inhibit the bipolar cell. In other words, to elicit a response from this ganglion cell, more light has to fall onto the ON area, that is the central area above, than its surrounding OFF area. This idea that adjacent areas of visual space produce opposite responses within a cell is known as lateral antagonism. Lateral antagonism is a processing strategy implemented throughout the brain. It's a strategy that highlights differences in inputs and minimizes redundancies. The brain is optimized to extract differences which exist in space and in time. In the movie we're about to see, we'll observe the mapping of ganglion cell receptive fields. The sounds you'll hear represent the electrical discharges or the firing rate of the cell. As you'll see, these ganglion cells do not respond well to large fields of light. Instead, they require variations in light across their receptive fields. Ganglion cell receptive fields also vary in size. Small receptive fields, such as those found in the fovea, encode high resolution image detail. Larger receptive fields, such as those found in the periphery, encode low resolution blurry image structure. So why does peripheral vision afford us poorer resolution than central vision? The first answer to this has to do with the higher density of cones present in the fovea relative to the periphery, where rods are more numerous. The second reason has to do with the greater degree of convergence in the periphery. Finally, there is another reason, a phenomenon known as visual crowding. Crowding refers to the inability to identify peripherally presented objects due to nearby clutter. You can see this for yourself. If you stare at the blue cross, you probably have no problem identifying the letter immediately to its right. If, however, you now look at the red cross, that same letter is much more difficult to identify when presented in the sequence of letters that are close to it. This is crowding, and it's something we'll explore in detail next week.